In this video, we are going to learn about topics related to transport of mRNA or transport of RNAs from nucleus to cytoplasm, then genetic translation, and some other related topics. So, in eukaryotes, mature RNAs are transported from nucleus to cytoplasm as complexes with proteins, as ribonucleoproteins. This complexing is essential since the associated proteins not only facilitate the transport but also provide additional protection from the enzymatic breakdown of RNAs. Usually, mRNA binds with a protein called information carrier. Informopher. It's a protein. On reaching the cytoplasm, the mRNA informopher complex binds with some cytoplasmic proteins and form an informosome. This in turn binds with ribosomal subunits and forms an mRNA ribosome complex. So, mRNA binds with a protein called informopher, and after that, when that particular structure reaches the cytoplasm, the mRNA informopher complex binds with some cytoplasmic proteins and then forms informosome. This in turn binds with ribosomal subunits and forms an mRNA ribosome complex. Now, the major topic, genetic translation or protein biosynthesis. Genetic translation or protein synthesis is the final stage of gene expression. During it, the genetic message programmed or transcribed in mRNA from DNA in the form of ribonucleotide sequence is used to specify the amino acid sequence of protein. So it would be transferred to protein in the form of amino acid sequence. This is accomplished by mRNA dependent synthesis of protein. Then mechanism of protein synthesis. Protein synthesis essentially involves the activation and assembling of amino acids in a specific sequence as per the instructions laid down in the genetic code. Protein synthesis eventually involves the activation and assembling of amino acids in a specific sequence as per the instructions laid down in the genetic code. The modulator machinery of protein synthesis includes RNAs, ribosomes, activated tRNA bound amino acids, and a variety of accessory factors such as enzymes, cofactors, enzyme rich compounds, and metallic ions. Messenger RNA serve as the template for protein synthesis. It has an open reading frame and a ribosome binding site. The reading frame is open because it is formed of a sequence of only translatable exons not interrupted by introns. The binding site is the site where the small ribosomal subunit binds and the rest of the ribosome is assembled during protein synthesis. Transfer RNA has a viral role in genetic translation. It functions as a versatile adapter molecule to carry activated amino acids from their cytoplasmic pool to the protein synthetic site in ribosome exactly in the correct order specified by the mRNA sequence. This ensures the amino acids are assembled and joined together in the correct sequence. Cells usually contain 31 to 40 different kinds of tRNAs. Each of them binds only to one of the 20 kinds of amino acids. So there will be more than one kind of tRNAs for each amino acid. The different kinds of tRNAs which bind to the same kind of amino acids are called isoacceptor tRNAs. Isoacceptor tRNAs. Ribosomes are the centers of protein synthesis. Their different domains have important roles in protein synthesis. A eukaryotic ribosome has typically three major binding sites, namely M site or mRNA, A site, aminoacyl tRNA site, or the incoming aminoacyl charged tRNA, and P site, peptidyl tRNA site, or 4-peptidyl tRNA. Bacterial ribosome has yet another site also, namely E site or exit site, to which the spent tRNAs, deacylated or deaminated tRNAs, are dispatched before their ejection. The later ribosomal subunit has a peptidyl transferase domain and GTPase domain. The former serves as a catalytic site in peptide bond formation, and the later plays a significant role in the translocation of ribosomes along mRNA. Some accessory factors associate with ribosome only transiently. They have significant role in the assembly, disassembly, and functional activity of ribosome. The energy sources of protein synthesis include ATP and GTP. Ribosomes serve as the centers of protein production. They read the coded information in the mRNA and accordingly assemble amino acids in a specific sequence to form a protein. mRNA acts as an intermediate molecule in the translation of nucleotide sequence of DNA to the amino acid sequence of protein. During this, nucleotide codons are converted to amino acid sequences. tRNA acts as an adapter molecule which binds with activated amino acids and orients them on the mRNA template in the correct sequence. The protein factors involved in genetic translation include initiation factors, elongation factors, and termination factors. Initiation factors are involved in the initiation of translation by incorporating the first amino acid. Elongation factors bring about the movement of ribosome along mRNA and the elongation of the polypeptide chain by the incorporation of more and more amino acids. Termination factors recognize the stop codon on the mRNA and terminate chain elongation and protein synthesis. Protein synthesis is a complex and multi-stage process. Its major stages include recognition and ATP-dependent enzymatic activation of amino acids, adaptation of activated amino acids by tRNA, formation of mRNA ribosome complex, movement of the activated and adapted tRNA-bound amino acids to polysome, tRNA mRNA binding and codon-anticodon pairing, then translation of the genetic code and synthesis of polypeptide chain. First step, recognition, activation and adaptation of amino acids. For protein synthesis, amino acids are selected from the amino acid pool of cytoplasm. For protein synthesis, amino acids are selected from the amino acid pool of cytoplasm. Before assembling to proteins, they get activated or energized by reacting with ATP and the activating enzyme aminoacyl tRNA synthetase. During this, the required amino acids are recognized and selected by the enzyme. All the known proteogenic amino acids and the isomers of proteogenic ones are rejected in this process. Each selected amino acid soon reacts with ATP and the enzyme and forms aminoacyl adenine synthetase, aminoacyl adenylate enzyme complex. This enzyme bound aminoacyl adenylate is the activated form of amino acid. Each kind of amino acid is activated by a specific enzyme. So there will be at least 20 kinds of aminoacyl tRNA synthetase. Each activated amino acid soon covalently links with the 3' end of a specific tRNA through an enzyme-rich ester bond forming aminoacyl tRNA complex. This is known as adaptation of amino acid, charging of tRNA, or aminoacylation of tRNA. This is also mediated by the activating enzyme. During adaptation, the enzyme parts with aminoacyl adenylate complex. Soon tRNA binds with the complex through ester bonding and forms aminoacyl tRNA complex, releasing AMP and the enzyme. So amino acid plus enzyme, aminoacyl adenylate synthetase, AMP enzyme complex plus PPI. AA amino acid AMP enzyme complex plus tRNA, aminoacyl tRNA plus AMP plus enzyme. Then next step, codon recognition and codon anticodon pairing. First step was recognition, activation and adaptation of amino acid. Next step, codon recognition and codon anticodon pairing. Soon after adaptation, the aminoacyl tRNA complex moves to the mRNA ribosome complex. Soon after adaptation, the aminoacyl tRNA complex moves to the mRNA ribosome complex by simple diffusion. On reaching the ribosome, it reads the genetic text of the mRNA, recognizes its specific binding site, and accordingly binds with an appropriate codon of mRNA through complementary base pairing by hydrogen bonding. The nucleotide triplet of tRNA that binds with the tip triplet codon of mRNA is known as anticodon. A codon and its anticodon will be complementary to each other in base sequence. This tRNA serves as an intermediator called adapter molecule for positioning the amino acids in the exact sequence in a growing polypeptide chain. This ensures that the amino acid sequence specified by the mRNA is most faithfully translated. Then genetic translation. Next step, genetic translation. Genetic translation is the transfer of the transcribed or programmed information from mRNA to polypeptide chain. The mechanism of translation in prokaryotes is similar to that of eukaryotes. During translation, the 4-letter nucleic acid language or nucleotide language is translated to the 20-letter protein language or amino acid language. Precisely, translation represents the actual process of protein synthesis. Then codon anticodon pairing, which brings about the orderly orientation of amino acids to form a particular protein, is the core process of translation. Then translation occurs on polysome, which is the active participation of Mg with the active participation of Mg tuples and a variety of enzymes and protein factors utilizing GTP and ATP as a source of energy. GTP is used for ribosome movement and the binding of accessory factors, and ATP is used for removing the secondary structure from mRNA. Translation is completed in 3 stages as in transcription, namely initiation, elongation, and termination. Initiation is the starting of peptide chain synthesis, elongation is the lengthening of the peptide chain, and termination is the end of peptide chain elongation. Initiation involves the binding of ribosome to mRNA and the incorporation of the first amino acid. Elongation involves the repeated addition of amino acids to the growing peptide chain. And termination involves the cessation of elongation of this release of nascent polypeptide chain. Then first step in detail, like initiation. Initiation involves the incorporation of the first amino acid of the polypeptide chain. So it includes all the preliminary reactions occurring before the formation of the first peptide bond between first and second amino acids. The significant events of initiation include the assembling of ribosome on mRNA, the positioning of ribosome at the translation start site, the assembling of molecular machinery called initiation complex, and the placement of the amino acids of polypeptide chain. The initiation complex is formed by a bonding between mRNA, ribosomal subunit, initiated tRNA, GTP, and a variety of proteins called initiation factors or IF. The presence of Mg2+ is necessary for this complexing. The presence of Mg2+ is necessary for this complexing. In bacteria, there are only 3 initiation factors, IF1, IF2, IF3. But in eukaryotes, there are at least 9 or 10 different initiation factors. IF1, IF2, IF3, IF4A, IF4B, IF4C, IF4D, IF4F, IF5, IF6. The formation of the initiation complex is relatively simple in prokaryotes than in eukaryotes. In prokaryotes, it is completed in the following successive steps. To begin with, IF2 binds with GTP, then IF1 and IF3 bind with a free 30S small ribosomal subunit. This inhibits the bonding between the large 50S and small ribosomal subunits and thereby prevents the formation of an mRNA-free inactive ribosome. The IF2 and GTP complex now binds with a small ribosomal subunit. This probably helps the subsequent binding of the charged initiator tRNA with the 30S subunit. Soon, the 30S subunit reaches to an mRNA, taking use of ribosome binding site on the mRNA. Then the initiator mRNA binds with the initiation codon of mRNA by codon anticodon pairing. The whole complex formed by mRNA initiated tRNA, 30S ribosomal subunit, IF1, IF2, IF3 and GTP are called primary or 30S initiation complex. So shortly afterwards, IF3 is released. Lastly, the 30S ribosomal subunit binds with the primary initiation complex, displacing IF1 and IF2. This forms the secondary or 70S initiation complex. This is an energy consuming step, and so GTP is hydrolyzed at this stage. In the secondary initiation complex, the ribosome has two tRNA binding sites, namely A site, acceptor site or aminoacyl site, and P site, peptidyl site or polymerization site. A site is the binding site for all incoming aminoacyl tRNAs, excepting the initiator tRNA. On the other hand, P site is the binding site for the initiator tRNA and all peptidyl tRNAs with an activated peptide fragment. Apart from this, there may be an E site or exit site for the expulsion of the unloaded tRNA. The binding of the smaller ribosomal subunit with mRNA is perhaps the most significant step of initiation. It is governed by a special signal codon of mRNA known as initiation codon or AUG. The second major event is the binding of the initiator tRNA with the initiation codon. Initiator tRNA is the first incoming aminoacyl tRNA that binds with the initiation codon. In the subsequent incoming aminoacyl tRNAs are called elongator tRNA. Since the initiation codon has AUG sequence, the anticodon of the initiator tRNA invariably will have UAC sequence. Characteristically, the amino acid linked with the initiator tRNA is always methionine. So the initiator tRNA is always methionine tRNA, and the first amino acid to be incorporated in all cases of protein synthesis is methionine. In prokaryotes attached to methionine, a 4-mile like CHO group. So in them, the initiator tRNA is 4-mile methionine tRNA. For the detection of the initiation codon, prokaryotes and eukaryotes use different signals. In bacterial mRNA, there is a specific non-translated leader sequence before the initiator codon. It is called Shine-Dalgrano sequence. It aligns itself with the complementary sequence of the 16S rRNA of the ribosome. In eukaryotic mRNA, a leader sequence equivalent to Shine-Dalgero sequence is altogether absent. So the ribosome recognizes the initiator codon of mRNA by scanning the mRNA from the 5' terminus until it comes across with the initiation codon AUG. Then next, elongation process. Elongation is the lengthening or polymerization of the peptide chain by the successive incorporation of more and more amino acids. It is mediated by certain protein factors known as elongation factors. Elongation includes all the reactions from the formation of the first peptide bond. It is mediated by certain protein factors known as elongation factors. Elongation includes all the reactions from the formation of the first polypeptide bond up to the incorporation of the last amino acid. During this, there is the movement of translocation or translocation of ribosome along the mRNA in convoys from the initiator codon to the termination codon. It is assumed that GTP hydrolysis provides energy from conformational changes in ribosome, which in turn facilitates translocation. Elongation involves the following 5 major events: binding of all the incoming aminoacyl tRNAs to A site. First step, binding of all the incoming aminoacyl tRNA to A site. This may be called aminoacyl tRNA delivery. Then binding of peptidyl tRNA to P site. Transfer of the peptidyl residue or attached peptide chain from the peptidyl tRNA in the P site to the aminoacyl tRNA in the A site. This is called transpeptidation. Then peptide bonding between the amino acyl residue of the amino acyl tRNA and the newly added peptidyl fragment. Transpeptidation and peptide bonding are mediated by the enzyme peptidyl transferase or peptide synthetase. This enzyme works in collaboration with another one called tRNA deacylase, which breaks the link between methionine and its tRNA after the formation of peptide bond. Expulsion of the unloaded or peptidyl-free tRNA from the P site to the outside. This is affected by ribosome movement. The cyclic repetition of all the above processes over and over again brings about the successive addition of amino acids to the growing peptide chain. To begin with, the initiated tRNA, methionine tRNA, binds at the P site during initiation. Then the second incoming amino acyl tRNA binds at the A site as specified by the mRNA codon, which is next to the initiation codon. This is the first step of elongation. Soon methionine from the former is transferred to the later, transpeptidation, and gets peptide bonded to the later amino acyl residue. As a result, dipeptide is formed. The whole process is mediated by the enzyme peptide synthetase or peptidyl transferase, which works in conjunction with another enzyme called tRNA deacylase. The latter breaks the linkage between methionine and the initiated tRNA. Now ribosome moves toward the termination codon. As a result, the unloaded or uncharged initiated tRNA gets pushed out from the P site, and the next time the second tRNA, now with the attached peptidyl residue, comes to occupy the P site. Soon the third amino acyl tRNA comes in and occupies the A site, and the whole cycle of events is repeated, forming a tripeptide. In this manner, amino acids are added to the growing peptide chain one by one. This is called peptide chain elongation. As a result of it, the whole message encoded by mRNA gets sequentially transferred to protein. This, in turn, brings about gene action or gene expression. Then, termination. Translation termination is the cessation of peptide chain elongation and the subsequent release of completed chain. So, it involves all the events occurring after the formation of the last peptide bond and therefore the detachment of the mRNA. It is signaled by a termination codon or stop codon UAA, UAG or UGA of mRNA. Termination takes place when the ribosome encounters the stop codon, which is now positioned at the A site. Termination is mediated by certain proteins known as release factors or termination factors. They help ribosome to recognize a stop codon. Since stop codon do not code for any amino acid, no tRNA species will have an anticodon complementary to a stop codon. So no tRNA can recognize the stop codon. On the other hand, the ERF or release factors can recognize it and bind with it. Termination consists of the following major steps. The release factors first recognize the termination codon in the A site and bind with it. Soon the release factor interact with the peptide synthetase and make it hydrolytic. Hydrolytic peptide synthetase now hydrolyze the ester bond between the last tRNA and the last amino acid of the peptide chain. As a result, the completed polypeptide chain is released from the ribosome. Methionine, which is the first amino acid incorporated, is soon removed from the polypeptide chain by the action of peptidases. The last tRNA is soon expelled from the P site to the outside. Soon the ribosome dissociates from the mRNA as subunits. These subunits can re-enter the ribosome cycle for the synthesis of another protein molecule. The detached tRNA can pick up another amino acid and continue its activity. But the detached mRNA is hydrolyzed to ribonucleotide units by ribonucleases. Then next major topic is about post-translational protein modification, which will be explained with the help of next video. Thank you for listening this video.